Hi, welcome to this unit on psycholinguistic and neurolinguistic methods, part of the course on research methods in linguistics. The questions we care about in language science include what is language, how is it organized as a system in the mind or brain, how did it evolve for our species, how does it vary over space and time, how is it learned or acquired, and how does it develop in children, and finally, and that is our focus today, how is it processed? Answering these questions will obviously require different kinds of theories, different methods, and different data types. Together, this form a complex landscape that is growing and diversifying a little bit more every day. Today, I will give you an overview of parts of this landscape, specifically when it comes to methods for studying language processing experimentally. Psycholinguistics and neurolinguistics focus on processes that happen at fairly short timescales, in the order of milliseconds to seconds, and we will see a few examples later. Understanding these processes has implications also for more extended processes like language evolution and language development, as well as more indirectly for language structure. So we can use information that we gain from experiments on language processing to say something about all these other aspects of language, including our knowledge of language. Let us now turn to methods. We know that research involves the formulation, testing, and modification of theories or hypotheses based on observation or experiment. Now, this process is not free, but is regimented by methods, which tell us what moves are allowed in the game of testing and revising our hypotheses. Methods then are validated, controlled, repeatable research procedures. In psychoneurolinguistics, research methods typically include procedures for designing research, for example, for designing experiments or surveys or field studies. They also involve instruments and procedures for data collection, as well as mathematical procedures for data analysis, such as statistics. Now, methods are not fixed forever. They too evolve, but they evolve more slowly compared to the rate at which new empirical results appear. One important idea that I'd like to convey to you is methodological pluralism. This quote by Ambridge and Leven is one of many that express the idea. They write that since it is, of course, impossible to investigate speakers' knowledge of language directly, then various methodologies are required in order to infer this knowledge from observable behavior, for example, from experimental data. It is important to bear in mind that no single method provides a pure measure of linguistic knowledge. Pluralism, then, is the view that we must integrate, combine different methods, to use the data that we collect using these methods, and to draw inferences about how language is processed and organized. Now, I cannot review the entire history of psycholinguistics or neurolinguistics here. Those of you who will take the language and cognition course that I teach in the spring will learn more about the history of these fields. What I want to do today is just to highlight two important moments in the development of these fields when the importance of empirical observation and experimental data became codified in their methodologies. One important step was due to the French neurologist Paul Broca around 1861. So Broca discovered that lesions to particular parts or areas of the brain can result in speech deficits. The images you see here show the left hemisphere of two patients, Le Borgne, who died at age 51, and Lelong, who died when he was 84. Now, both patients have visible lesions in the left inferior frontal cortex. So that is the anterior part of the left hemisphere of the brain. Now, these patients were only able to utter a limited number of monosyllabic, very high frequency words in French, as is the case for Lelong here. Oui, non, toi, toujours, and his own name, Lelo. Or the single syllable, tan, for Le Borgne. Now, Broca concluded from examining these two patients that this lesion caused the speech deficit. So Broca helped establish the methodological principle that clinical data about brain lesions and the cognitive deficits that they are accompanied by can provide information about where certain functions may be localized in the brain, or also what parts of the brain are important for the normal execution of those functions. And clinical data are, of course, still in use and still very important today. The second major turning point in the history of psycholinguistics and neurolinguistics is due to Willem Wundt, who was responsible for introducing the experimental method into the study of the mind around the turn of the 20th century. In his book, Outlines of Psychology, Wundt says that in psychology, exact observation is possible only in the form of experimental observation, and that psychology can never be a pure science of observation. By this he means sciences like geology, where we can observe rocks, fossils, and so on, or astronomy, where we can point a telescope and actually see celestial objects. Wundt says that psychology is not like those sciences. He writes that the contents of this science, psychology, are exclusively processes and not permanent objects. So to investigate with precision how these processes rise and progress over time, how they are composed out of simple component processes, and how these simple processes interact, all of this is possible only through experiment, not through introspection. So to put it crudely, you cannot look inside yourself and hope to find the objects of psychological science available for observation. So in the 20th century, researchers started developing more sophisticated experimental methods for studying language and other mental processes. One of these methods is eye tracking, where we use an infrared camera to track the movements of the pupil. Here you see a simplified representation of eye tracking data for a participant reading the sentence, "The knight attacked the windmill on his donkey." The data contain information about fixations, so the radius of the pink circles represent the time spent looking at a specific words, saccades, rapid automatic jump-like movements of the eyes to the next word, and regressions, where the eyes move back to words that have already been fixated at. So there is a lot of information in the data. For example, we see that content words, like nouns and the verb, tend to have longer fixation times than determiners. Also, determiners appear to be skipped. In fact, they are read based on information in the visual periphery, while the participant fixates an adjacent word. Here's an exercise for you. Consider the eye movements for the sentence in the previous slide and try to provide an explanation in terms of language processes for the regression from the word windmill back to attacked. Try to imagine why the reader should have to look again at attacked and perhaps process it again, and what kind of process this might be. Eye tracking can be applied to a number of different tasks, not just to reading sentences, but also, for example, to inspecting visual scenes. This is an older study by Yarbus, which was taken to show that eye tracking is not as reliable as we may think, because depending on the instructions that we give to participants, their eye movements will look very different. For example, if the task is to give the ages of people in the scene, panel number 3, what the participant does is to look at the faces of people. Most eye movements are concentrated around the head of the people in the scene. Compare this to panel 5, for example, where the participant is asked to remember the clothes that are worn by the people in the scene. Now the eye movements might move up and down through the whole body of the people in the scene. The conclusion is that eye movements depend on cognitive goals. They are driven by internal representations, not just, and not even primarily, by the picture out there. In other words, the way you look depends on the goals you have. This also shows that eye movements are windows into cognition, not just into perception. Now, eye tracking has a number of assumptions that every experiment has to try to satisfy. If our question is how language is processed in real time, then we must make sure that eye tracking can tell us something about cognitive processes, not just visual perception. And we can do that if two assumptions are met. One is the strong eye-mind hypothesis, that there must be no significant or variable time lag between fixation, looking at something, and information processing. Because if there is such a time lag, then we cannot correlate eye movements with cognitive processes, so we cannot use the former to draw inferences about the latter. A second assumption is that all fixations should lead to information intake, and that the longer a participant fixates on an aspect of the stimulus, the more information is being gathered or processed about the stimulus. If not all fixations lead to information intake, or if there is no correlation between the duration of fixations and the amount of information processing, once again, it becomes difficult or impossible to use eye movements to draw inferences about cognitive processes. Eye tracking works only with experimental designs that maximize chances that these assumptions are valid. Now, all this reminds us that eye tracking is an indirect measure of cognitive processing. We know that cognitive processing happens in the brain, and ideally, we would like to be able to measure directly online activity in the brain that is correlated with cognitive processes. This we can do with a number of techniques, including EEG, which stands for electroencephalography. This involves measuring, in a safe and non-invasive manner, the electrical currents that are generated by the brain while, for example, we are reading sentences. We can manipulate semantic and grammatical aspects of the sentences, and we can study how the brain reacts to those manipulations. Already a century ago, it was observed by Hans Berger that the human EEG, so the electrical activity that the brain generates, is modulated by properties of external stimuli. For example, if you present participants with a light stimulus that is either always on or always off, and suddenly the light starts flickering, going on and off very quickly, then we see a significant change in the type of electrical activity that the brain produces. So this is shown at the top of the slide here. We go from essentially a slower, high amplitude oscillation to a smaller amplitude but faster frequency oscillation when the light starts flickering. What we do today is to run experiments where we present participants with sentences, typically word by word at a fixed rate, in the middle of a screen. And this is to avoid that participants move their eyes, as eye movements generate a lot of activity, largely produced by eye muscles, that cancels out the much weaker currents that we can pick up from the brain. So we then amplify the EEG signal and send the result to a computer to start processing the data. In order for us to be able to record EEG data at all, the brain has to produce electrical currents in large populations of neurons, oriented orthogonally with respect to the macroelectrodes that we put on the surface of participants' heads. Luckily, because of the anatomy of the brain, we are able to record quite a lot of the electrical activity that the brain is generating. One of the methods that we have to analyze EEG data is the event-related potential, or ERP, where we average signals from many different repetitions of the same event, which we call a trial, and over many participants in the experiment. By doing so, we greatly reduce the noise in the data, and we end up with a clean average signal, which is indeed the event-related potential. This study by Marta Kutas and Stephen Hilliard in 1980 is one of the first using ERPs in language processing. They had three conditions: control sentences like "it was his first day at work," violations of semantic expectancy like "he spread the warm bread with socks," and violations of perceptual expectancy like "she put on her high-heeled shoes." In a violation of semantic expectancy, we do not normally expect or predict the word socks to occur in the context of the preceding sentence fragment. He spread the warm bread with. We would expect something like butter or Nutella. In violations of perceptual expectancy, the word that closes these sentences, shoes, is presented in a larger font than we expect, given the font size of the preceding words. So they found two different effects. For the violation of semantic expectancy, they find an N400, a negative polarity brainwave peaking around 400 milliseconds, so less than half a second from the onset of the word socks. For the violation of perceptual expectancy, they found a P560, so this brainwave has a later peak at around 560 milliseconds compared to the N400. The fact that this is a positive polarity effect is evidence that different networks of the brain process semantic and perceptual information. What is also interesting here is that it takes less than half a second for the brain to start processing the meaning of a word in context. In a second experiment, Kutas and Hilliard manipulated the strength of the violation of semantic expectancy. They had moderate violations. For example, he took a sip from the waterfall. Here we would expect words like bottle or glass, but still waterfall kind of makes sense. We can integrate this word and interpret the meaning of the entire sentence. In contrast, in a strong violation of semantic expectancy, like he took a sip from the transmitter, we would not expect the word transmitter at all. We cannot interpret this sentence. They found that the strong violation of semantic expectancy results in a larger N400 compared to the moderate violation, which still results in an N400 effect, but a smaller one. That means that the N400 is not an all or none kind of response that the brain produces indifferently when there is a violation of semantic expectancy. Rather, the N400 is a graded response that depends on expectancy or the probability of a word given the semantics of the context in which it appears. EG2 has a number of assumptions that must be met if we want to be able to draw inferences about cognitive processes. One of these assumptions is that the processes of interest have to occur in well-defined time intervals that are moreover comparable across trials and participants. With EG, we can study processes that happen shortly after the presentation of a stimulus. Secondly, the processes of interest are revealed directly by comparing data between two or more conditions, let's say a violation of expectancy compared to a control condition, or a strong violation compared to a weak violation. The assumption of pure insertion means that the cognitive processes of interest can be revealed entirely by subtracting the brain activity of the first condition, which we assume does not engage the process of interest, from the activity of the second condition, which we assume does involve the process of interest. This simple logic is widely used, but does not always apply. So to use EEG and ERPs in their traditional form, we need experimental designs that maximize chances that these assumptions are valid. Another important point is that EEG works well with passive tasks, for example, reading or listening to sentences, but it does not work with active tasks, such as speaking. Again, the act of speaking engages many different muscles that have to control movements of the mouth and the vocal tract, and these muscles would generate electrical signals that interfere with the much weaker brain signals that we want to measure. EEG and ERPs are very good methods for tracking cognitive processes in real time, at timescales of fractions of a second. We say that these methods have good temporal resolution, but they do not have good spatial resolution. That is, we do not know where these signals are being generated in the brain. For that, we need a different set of methods. One of these is fMRI, which stands for Functional Magnetic Resonance Imaging. With fMRI, we are not looking at brain activity as such, but rather we are looking at blood metabolism, specifically blood flow, to restore oxygen to parts of the brain that have been recently active. With MRI, without the F for functional, we can look at structural properties of the brain. We can perform structural scans that are similar, but better, to computer tomography or even X-rays. MRI has largely replaced those older technologies because it has a much better spatial resolution. It can differentiate details in different parts of brain anatomy that we could not resolve before. This is a study where Nina Dronkers and colleagues have put the brains of Broca's 2 patients in an MR scanner, and they have been able to see what Broca was not able to see. Not having dissected his patients' brains, Broca was only able to see the superficial lesions in the left inferior frontal cortex. But with MRI, we're now also able to see anomalies in the white matter tracts that connect the left inferior frontal cortex to other parts of the brain. And we can see this much better here. On the left side of the brain of Le Borgne, white matter is clearly deteriorated while it is well preserved on the right hemisphere. MRI comes with a bonus type of signal, and this is the functional signal, where we can measure blood supply to recently active areas of the brain that restore the normal oxygen levels. Researchers have exploited this functional signal to ask questions such as what parts of the brain are more active when we process language. This is an early study by Marcel Just and colleagues, which appeared in Science in 1996. They used 3 experimental conditions. They had active sentences, the reporter attacked the senator and admitted the error. That's the easiest to process and understand. They had subject relative sentences, the reporter who attacked the senator admitted the error, where it's the reporter who attacked the senator and admitted the error. So the meaning is the same as in the active sentence, but syntactic structure is more complex. And finally, they had object relative sentences, like the reporter who the senator attacked admitted the error. Here, the senator attacked the reporter and the reporter admitted the error. The object relative is the hardest to process among these 3 sentence types. We want to know whether there are areas of the brain that follow this complexity pattern and respond with more activity for object relatives, a little less for subject relatives, and less still for active sentences. And they find that indeed 2 areas respond following exactly this pattern, with object relatives leading to the greatest amount of activation in terms of number of active voxels or units of brain volume, followed by subject relatives and by active sentences. This pattern was observed in Broca's region in the left inferior frontal cortex, as well as in Wernicke's region in posterior superior temporal cortex. What is important to remember here is that these are 2 different regions that are distant from each other, and both are in the left hemisphere. Similar results come from a study by Dave Embick and colleagues published in 2000. They used sentences that contained either 1 or 2 spelling errors and 1 or 2 grammar errors. If you want, you can now pause the video to look at what these errors look like in these example sentences. They found 3 regions activated by both spelling and grammar errors. Again, Broca's region, Wernicke's region, and the angular and supramarginal gyri, all in the left hemisphere. Grammar errors tend to activate this brain network more than spelling errors do. Here we see the dynamics of activation over time. When a sentence with a spelling or a grammar error is presented, activation goes up and then down. And this happens in a coordinated, kappa way in all 3 areas. Grammar errors lead to slightly more activation compared to spelling errors in all 3 areas. This network in the left hemisphere responds similarly to spelling errors and to grammar errors. But again, the amount of activation, and so presumably the processing cost, is greater for grammar errors than for spelling errors. We have seen examples of experiments using EEG and earpiece and fMRI. This picture represents the spatial and temporal resolution of different methods to record brain activity. Earpiece, the light blue volume in the figure, has good temporal resolution, from milliseconds to seconds, but its spatial resolution is poor, in the order of a few centimeters. In most cases, we cannot use EEG to precisely localize effects in brain space. In contrast, fMRI, which is the yellow volume here, has poorer temporal resolution, from 1 second up to a few minutes, which means that we cannot resolve fast cognitive processes in time using fMRI, but we can use fMRI to resolve activity in brain space, down to a few millimeters. This again brings us back to our point about methodological pluralism. that we need to integrate data from different methods, including EEG and fMRI, to come up with a better picture of language processing in the brain. Here is another task for you. EEG, earpiece, and fMRI can provide us with reliable information about when and where, respectively, certain processes occur in the brain. But we are actually interested in how the brain processes language. So an important question is how we can bridge the gap between questions of spatial and temporal localization in the brain to questions of mechanism, of how the brain processes language. I want you to reflect about how we can do that and how spatial and temporal data can provide a basis for answering questions of mechanism. Tip: consider that with methods like EEG, we can determine the relative temporal order of processes, and with fMRI, we can determine whether processes recruit the same or different or overlapping brain networks. So how can we use this type of information to understand better how the brain processes language? To summarize what we covered today, we said that methods are supposed to give structure to the research process and to ensure the reliable generation of data that are relevant to our hypotheses. The goal is to draw valid inferences from the data that we collect to the cognitive and neural processes that enable language learning and use. In doing so, we can also gather indirect information about the mental structures that constitute our knowledge of language. We have also said that there is no single best method in psycholinguistics and neurolinguistics. There are preferred methods given certain questions, and each method has its own strengths and limitations. We have looked at three widely used methods: eye movements, event-related potentials, and functional brain imaging. We've also noted that they have complementary uses. They need to be integrated in the long run to arrive at more complete pictures of what happens in the brain in space and time, and from there to draw inferences about the cognitive mechanisms that support language learning and use. The recommended readings for this lecture are the first 3 chapters of my book Neurolinguistics, which you can find online from the NTNU library, and then 3 optional readings, one on eye tracking, one on event-related potentials, and one on functional imaging. Thank you for your attention.